Thank you very much for your kind introduction, Therese, and welcome everyone also from my side to today's webinar on the tryptophan metabolism at the intersection of immunity, neuronal function, and gut homeostasis. My name is Barbara Ustaszewski, and I'm a scientist at the Research and Development Department at Berkeley's Life Sciences based in Innsbruck, Austria. So what can you expect to hear in the next approximately 30 minutes? First, I will give you a brief introduction on the relevance of the tryptophan metabolism in health and disease. Then we will have a look at 3 use cases. This is cancer, inflammatory bowel disease, and neurological diseases. After that, I will introduce you to our tryptophan metabolism assay. And in the end, we will come, as always, to a short summary of today's webinar. So let's start with a short introduction. So the aromatic amino acid tryptophan is an essential amino acid which is mainly taken up via the diet, especially when protein-rich food like meat or dairy products, but it can also be produced to a certain extent from the gut microbiota. And as all proteinogenic amino acids, also tryptophan is probably best known for its role as a building block for the synthesis of proteins. But far less known, but at least equally important, is the metabolization of tryptophan into several bioactive molecules. And this is facilitated by 3 main catabolic pathways: the serotonin, the kynurenine, and the indole pathway. So let's start with the serotonin pathway. So 1 to 10% of the dietary tryptophan is metabolized via the serotonin pathway. And serotonin is a neurotransmitter, and about 95% of serotonin is actually synthesized in our gut epithelium by specialized cells located there. So knowing this, it is not very surprising that also the gut microbes can modulate the availability of serotonin. So the physiological effects of serotonin are that it is a key molecule for signaling along the gut-brain axis. It is central in the central nervous system. It exerts positive effects on mood, sleep, and behavior. It also improves memory and gut motility. Then up to 95%, so the vast majority of dietary tryptophan is metabolized along the kynurenine pathway, and this pathway can be highly induced by inflammation, and its activation leads to an anti-inflammatory response. So several metabolites of this pathway exert their immune modulatory effects by activating a nuclear receptor, the so-called aryl hydrocarbon receptor, or AHR. Other metabolites regulate the carbohydrate metabolism, and some are involved in the regulation of the energy metabolism. And the third route for tryptophan catabolization is the indole pathway. Along this pathway, various intestinal bacteria metabolize tryptophan into indole and its derivatives. These bacterial metabolites all impact the host immune system. They can therefore also be understood as interkingdom signaling molecules of the gut immune axis. Some of these can again activate the AHR, which is generally thought to reduce inflammation in order to maintain gut homeostasis. And other metabolites again bind to another nuclear receptor, the so-called pregnan X receptor, or PXR, to maintain intestinal barrier function. So overall, we can say that tryptophan metabolism can be considered as an evolutionary strategy that links nutrition to systemic effects on health and disease. So let's have a closer look at the metabolites involved in these 3 pathways of tryptophan breakdown. The first step in the synthesis of the neurotransmitter serotonin from tryptophan is carried out by the rate-limiting enzyme tryptophan hydroxylase, or TPH. And as mentioned before, this happens mainly in the gut, but some serotonin is also produced directly in the brain. And what I would like to point out here is that the enzymatic activity of TPH can be estimated by the calculation of a ratio of serotonin to tryptophan. And the higher the ratio, the higher the activity of the TPH enzyme. Serotonin can then be converted into the hormone melatonin or to 5-hydroxyindolacetic acid, which is rapidly excreted in urine. Now let's move on to the kynurenine pathway. Kynurenine is formed from tryptophan by the rate-limiting enzyme either indolamine 2,3 dioxygenase or IDO, which is expressed in all tissues, or tryptophan 2,3 dioxygenase or TDO, which is mainly expressed in the liver. And again, here the enzymatic activity can be inferred by the ratio of kynurenine to tryptophan. And the higher the ratio, the higher the IDO or TDO activity. Then kynurenine is mainly converted either by the enzyme kynurenine aminotransferase or CAT along the kynureninic acid arm, or by the enzyme kynurenine monooxygenase KMO along the quinolinic acid arm. The kynureninic acid arm has anti-inflammatory and neuroprotective properties, as the metabolites generated by this arm can bind to the nuclear receptor AHR, while the quinolinic acid arm has pro-inflammatory and rather neurotoxic properties. But quinolinic acid can also serve as a precursor for NADH+, which is a crucial coenzyme in energy metabolism. NAD+ is actually mainly generated from nicotinamide and nicotinic acid, which is also known as vitamin B3. And as mentioned before, the third main option for tryptophan catabolism is mediated via gut bacteria, and some of them can convert tryptophan into indole and indole derivatives like 3-indole propionic acid. And 3-indole propionic acid binds, for example, to the PXR receptor and to exert anti-inflammatory effects. So overall, it is a well-regulated and balanced tryptophan metabolism is crucial to maintain health, and it is involved in a multitude of important physiological processes such as immunity, neuronal function, and gut homeostasis. The tryptophan metabolism can be indeed deregulated, for example, by inflammation or dysbiosis. This is an imbalance in the gut microbiota. And the deregulation of one pathway also results in a deregulation of the two other pathways, since they are all interconnected via tryptophan availability. And if this unfavorable deregulated condition persists or is too severe, this can result in a wide variety of diseases. For example, metabolic diseases like obesity or diabetes, or cardiovascular diseases like coronary heart disease or stroke, but also autoimmune diseases like multiple sclerosis or allergies. Interestingly, also infections can impact tryptophan metabolism like influenza A or pneumonia. But today we will focus mainly on cancer, on gastrointestinal disorders, and on neurological disorders. But before we come to that, I'd like to mention that there are multiple target sites in the tryptophan metabolism for therapeutic interventions for all kinds of diseases. So basically, there are 6 different therapeutic strategies currently in use or under development. First of all, the administration of tryptophan or tryptophan metabolites has a positive effect, for example, on the mood, on behavior, on sleep, or also on neuropsychiatric disorders like depression. Then also the use of pre- and probiotics is a promising therapeutic approach, as we have seen that the gut bacteria are tightly interconnected with the tryptophan metabolism. For example, the administration of lactobacillus to patients suffering from depression has been shown to improve cognitive function in these patients by lowering kynurenine levels in plasma. Then we have, of course, the well-known small molecule inhibitors, of the block enzymes such as IDO, TDO, KMO, but also receptors like the HR. Besides that, there are also antibodies that are used to block, for example, enzymatic activities. There are cell-based therapies currently being explored, especially for autoimmune diseases. And there are gene therapy approaches on the way, for instance, for delivering key enzymes to a specific site by the use of viral vectors. In this schematic representation of the kynurenine pathway, we can see the different key enzymes that are targeted by small molecule inhibitors. So IDO or TDO inhibitors, used primarily to treat cancer or neurological diseases, completely block the kynurenine pathway as they target the very first and rate-limiting step in this pathway. And this can in turn also boost the serotonin production. KMO inhibitors only block the pro-inflammatory neurotoxic quinolinic acid arm, which leads to an increased production of the anti-inflammatory neuroprotective metabolites of the kynureninic acid arm. And there are also CAT inhibitors that would block the kynureninic acid arm and boost rather the quinolinic acid arm. So you can see, depending on the specific context of the disease, which is important to know, you need to correctly choose the one or the other therapeutic approach. And this is also why experts from the field recommend a more complete and more comprehensive and also a tissue-specific analysis of tryptophan metabolites to better understand the overall role of the tryptophan metabolism in health and disease, and which is required for a better selection of therapy approaches. So now let's have a look at 3 use cases. So in the case of cancer, tumor cells frequently overexpress IDO, which also correlates with a poor clinical prognosis in many different cancer types. But why is that? So tumor cells overexpress IDO, which supports inflammatory processes in the tumor microenvironment, so that also dendritic cells residing there activate IDO. This activates T regulatory cells, which mediate immune tolerance to tumor antigens and suppress anti-tumor immune response by inhibition of cytotoxic T cells and natural killer cells. So together, IDO activation is a mechanism frequently activated by the tumor to escape immune surveillance by generation of an immune suppressive microenvironment, which helps the tumor cell to survive, which may lead to cancer development and progression and eventually to metastasis. So this is why the inhibition of the rate-limiting enzyme IDO is a very promising therapeutic target in cancer. Several studies have shown that, for example, patients with hematological cancers or colon cancer have reduced tryptophan levels and elevated kynurenine levels, so also an elevated kynurenine-tryptophan ratio, which reflects an enhanced IDO activity. Interestingly, also the anti-inflammatory kynurenic acid and quinaldic acid are also elevated in this setting. And upon remission, the kynurenine-tryptophan ratio drops, which might therefore serve as a prognostic marker. Another study by Miolo and colleagues has proven that reduced tryptophan and kynurenine levels in the serum of HER2-positive breast cancer patients predict a positive response to a neoadjuvant therapy. In this case, they administered or applied trastuzumab paclitaxel therapy. But there is also, besides this predictive marker potential, there is also a mechanistic explanation why responders have reduced tryptophan levels compared to non-responders. And this is since trastuzumab is thought to confer not only cytostatic effects through blocking HER2, but it is also thought to exert immunocytotoxic side effects by activating T cells and natural killer cells. So those patients who do not have an activated kynurenine pathway in the tumor microenvironment, so that would generate an immune suppressive environment for the tumor, they better respond to the administered treatment. So these are all nice examples for the predictive and prognostic potential of tryptophan and the tryptophan metabolites, which is the basis for the development of companion diagnostic tests. And lowering the kynurenine tryptophan ratio by, for example, IDO inhibitors is a promising therapeutic approach in cancer that is currently assessed in a multitude of clinical trials, especially in combination with other therapies like chemotherapies or immunotherapies. So let's have a look at the situation in gastroenterological diseases. So here, especially the kynurenine pathway is activated by unfavorable conditions like inflammation or dysbiosis. As said before, since pro-inflammatory cytokines, especially interferon gamma, but also TNF-alpha or interleukin-6, for example, induce the expression of the key enzyme IDO. So the majority of the available tryptophan is funneled into the kynurenine pathway with the intention to induce anti-inflammatory effects, but which can lead to the unintended accumulation of pro-inflammatory metabolites if the condition is too severe or persists for a too long time. At the same time, there is less tryptophan available for the indole pathway, which leads to a reduced production of indole metabolites that are important for the protection and regeneration of the intestinal mucosa, which then results in an increased gut permeability. And this again causes inflammation, which activates the kynurenine pathway and which can be seen as a vicious circle. And of course, the tryptophan deficiency also leads to a reduced serotonin production, which may result in mood problems, anxiety, or depression. This is also why many patients with gastrointestinal disorders like irritable bowel syndrome or inflammatory bowel disease frequently display neuropsychiatric comorbidities such as depression. But also the other way around, like patients with neurological disorders often present digestive disorders. And besides a continuous better understanding of the cause and the pathophysiological context of these diseases, many patients do not respond satisfactorily to approved medications or develop severe side effects. So hence there is an urgent need for the development of prognostic and predictive markers as well as additional therapeutic approaches, which is very promising here for inflammatory bowel disease. One example I'd like to show you here in which tryptophan and its metabolites correlate with disease activity and therapy response is a study from a German research group where they investigated the metabolite profiles of serum samples from over 500 patients with either ulcerative colitis or Crohn's disease, which are the two major forms of inflammatory bowel disease. And what they found was that they found decreased tryptophan levels and increased kynurenine levels, resulting in an increased kynurenine tryptophan ratio, indicating an enhanced IDO activity. They also found reduced levels of kynurenic acid and increased levels of quinolinic acid in IBD patients compared to controls. And this was even more pronounced with increasing disease activity and increasing levels of C-reactive protein, which is an established biomarker for chronically active inflammation. And besides that, they have also studied tryptophan levels upon treatment of the IBD patients with the standard therapeutic agent infliximab, which is a monoclonal antibody that blocks the pro-inflammatory cytokine TNF-alpha. And what it found was that in patients that respond to the therapy seen in figure A, the serum tryptophan levels stay high at least up to 24 weeks. While in non-responders, depicted to the right, tryptophan levels do rise in the beginning, but then decline again back to the low level before treatment. So this is a nice example for a predictive marker, potentially. In other studies, low level of indole propionic acid were detected in serum from patients with ulcerative colitis, which nicely fits into the picture since the majority of the tryptophan is metabolized via the kynurenine pathway. There's nothing left for the indole pathway. And in line with that, microbes metabolizing tryptophan are also diminished in IBD patients. This was found in one study. And another recent study has shown that the administration actually of indole propionic acid indeed improves gut barrier formation and ameliorates the disease. At least this was shown in mice. It still needs to be proven in humans. So these are all examples that have shown that tryptophan and its metabolites serve both as prognostic and predictive biomarkers, as well as therapeutic targets in gastroenterological diseases like inflammatory bowel disease. So our third example are neurological diseases, and here we have basically the same situation that we have seen before, that is systemic activation of IDO via pro-inflammatory cytokines. But here the enzyme kynurenine monooxygenase, KMO, plays an important role as it converts kynurenine along the quinolinic acid arm into neurotoxic metabolites. And together with the parallel reduction of the neuroprotective metabolites of the kynureninic acid arm, this may cause excitotoxicity, which is the damage or death or even death of neurons due to an excessive stimulation by neurotransmitters. And this can eventually result in neuropsychiatric disorders like depression or neurodegenerative diseases like Alzheimer's disease. So a balance between the neuroprotective kynurenic acid and the neurotoxic quinolinic acid is important for health, as we will see later. So collectively, both IDO and KMO represent a promising therapeutic target for the treatment of neurological disorders. So in neurological diseases, both neuropsychiatric disorders like depression or schizophrenia, but also neurodegenerative diseases like Alzheimer's or Parkinson's, quinolinic acid levels are frequently high, while kynurenic acid levels are observed to be frequently low in plasma and serum from these patients. And this has been shown to have also a prognostic value. For example, in a recent study by Chang and colleagues, they observed reduced levels of the ratio of kynurenic acid to kynurenine, which reflects the neuroprotective kynurenic acid arm in plasma from Parkinson's disease patients compared to normal controls. And at the same time, they saw a shift towards the neurotoxic quinolinic arm, reflected by the ratio of quinolinic acid to kynurenic acid. But also the kynurenine tryptophan ratio has been shown to play a role here. It was shown that an increased kynurenine tryptophan ratio in serum from patients with depression predicts a successful response to an anti-inflammatory add-on treatment. So those patients who have already an activated kynurenine pathway as an anti-inflammatory response profit more from this anti-inflammatory therapy. And in a study led by the National Institute of Health, it was further shown that ketamine, which is a drug that is frequently used for the treatment of depression, indeed decreases kynurenine levels in plasma. And by showing this, they could indeed uncover the so far unknown mechanism of action of this drug, having anti-inflammatory properties. In addition to that, both kynurenine and kynurenine tryptophan ratio were lower in responders than in non-responders, which makes them suitable as biomarkers to predict or monitor therapy response. So taken together, especially the administration of tryptophan metabolites and the use of KMO inhibitors are promising therapeutic strategies in neurological diseases. So now I'd like to introduce you to our tryptophan metabolism assay. So here are some key facts about the assay. It is a fee-for-service UHPLC MS-MS assay, which is performed in our service lab and has been fully validated in accordance with the FDA and EMA guidelines. The assay covers 17 tryptophan metabolites, which are absolutely quantified, and it requires a sample volume of 100 microliters. The absolute quantitation of the 17 tryptophan metabolites is achieved by a 7-point calibration, the use of internal standards, and also 3 quality controls in 3 concentration levels are used. So let's have a look at the metabolite coverage of the tryptophan metabolism assay. The assay provides a comprehensive coverage of all 3 major pathways of the tryptophan catabolism with a strong focus on the kynurenine pathway. In addition to that, the assay contains also the related metabolite neopterine. The production of neopterine is in certain immune cells like macrophages or dendritic cells is also stimulated by the cytokine interferon gamma, which, as we know now, induces at the same time the degradation of tryptophan. So in many cases, increased concentrations of neopterine correlate with decreased levels of tryptophan and increased levels of tryptophan degradation products. Neopterine is therefore an established marker for the activation of the cellular immune system. And it has been described to provide independent prognostic but also predictive value in many diseases ranging from viral infection to cardiovascular diseases and cancer. So, in line with the biocrates philosophy, also the tryptophan metabolism assay is a quality-controlled and standardized assay in order to deliver quantitative and reproducible results. And overall, with this assay, you can achieve great selectivity and high sample throughput. Here in this diagram, we can see the calibration range for each of the 17 metabolites represented by the purple bars. And each symbol depicted here is a different sample matrix. So besides human plasma and serum, we have also tested human fecal samples and urine, mouse plasma, and pig brain tissue homogenate. We have tried to set the 7-point calibration ranges so that they span across all kinds of sample matrices with a focus on human plasma, as you can see here, so that the vast majority of metabolites is above the limit of detection. So altogether, we can conclude here that the calibration ranges of the assay nicely fit to a broad range of sample matrices in which the metabolites can be comprehensively be detected and quantified. So with this, I'd like to summarize today's webinar. So we have seen that the tryptophan metabolism comprises 3 catabolic pathways that link nutrition to systemic effects on health and disease. Furthermore, it provides multiple promising target sites for drug development, and especially tissue-specific and a comprehensive analysis of tryptophan metabolites is recommended by experts from the field. And this is why we have developed the tryptophan metabolism assay, which is a standardized and quality-controlled assay following the biocrates philosophy with a comprehensive metabolite coverage applicable to various matrices and providing quantitative and reproducible results. So overall, the tool is a quantitative metabolomic solution at the intersection of immunity, neuronal function, and gut homeostasis. If you'd like to have more information on the tryptophan metabolism assay or our technology in general or other products, please just visit our webpage. And with this, I'd like to thank you for your attention. And if you have any questions or comments, please feel free to contact me via my email address directly or via LinkedIn or Twitter. Thanks again. Bye.